I'm greeting you from the conference room in my laboratory on the east side of Manhattan in New York City. And I send my greetings to all of you in the north of Norway. I wish I could be there, and it's my loss because, as some of you may know, I did postdoctoral work in Sweden in the 1960s, and I fell in love with Scandinavia, and I would very much like to be there, particularly as midsummer approaches. What I want to talk to you about is stress, and stress over the life course. And I want to give you a bit of background as to where I'm coming from, and this will make it easier to understand the story that I will then tell you. We discovered some years ago that this area called the hippocampus, a brain area shown here, is a target for stress and stress hormones. The hippocampus is important in remembering events in time and space. And in fact, the London cab driver study showed that when a cabbie remembers the route from one place to the other in London, his or her hippocampus becomes active. And in fact, the longer the cabbie is on the job as a cab driver, the larger that part of the brain becomes. So there's actually plasticity. The hippocampus is also important for food caching birds and squirrels in the wintertime, locating and retrieving food. And the hippocampus is also a target for sex hormones. And this has become of great interest in terms of postmenopausal hormone therapy and prevention of things like Alzheimer's disease. And finally, the hippocampus is sensitive to stress. We call it the canary in the coal mine of wear and tear on the brain associated with stress over the life course and during the aging process. Now, I want to give you a little bit of background about my career, so you'll, as I said, so you'll understand a little better what the rest of the talk is about. As a PhD student, I became interested in the study of gene expression, regulation of genes, and was introduced to what is now referred to as epigenetics, and we'll come back to that later. Worked with two scientists here at Rockefeller, Alfred Mirsky and Vincent Alfrey, who were pioneers in this area. My postdoctoral work in Sweden was with a cell biologist by the name of Holger Hudén, who introduced me to brain anatomy and function, and in particular to the hippocampus. And then when I came back to Rockefeller in the late 1960s, I joined the laboratory of the late Neil Miller, who is a pioneer in various aspects of psychology, but also in particular was one of the fathers of what is called behavioral medicine. And so this has helped to form who I am and what we have done. Now, what we found in the late 1960s is that the hippocampus, this is what it looks like in a rat, and this is a cross-section of the hippocampus, has receptors for stress hormones. They are shown here with the radioactive stress hormone, which exposed a film. And you can see at the low power all the major cell fields of the hippocampus, the dentate gyrus and Ammons horn, have these receptors. And at high power, you can see that many of them are in the cell nucleus. And in the cell nucleus, we know that these receptors have a biphasic effect. That is, low levels bind to one kind of receptor called a mineralocorticoid receptor and increase excitability, improve aspects of memory, but high levels of the adrenal steroid, a stress level, occupies another receptor and can have negative effects, impairing the same function. So we often talk about an inverted U-shaped dose response curve, and we'll hear a lot more about that later in the talk. When I had been doing this for a while at Rockefeller, I was introduced to the notion of interdisciplinary research through the late Elliott Steller at the University of Pennsylvania, and began to develop an interest in not only in medicine but also in the social sciences and how they interplay with brain science and with basic biology, neurobiology. And one of the organizations I became a member of, well, several organizations, were MacArthur Foundation networks, first on health and behavior. I was never a member of Successful Aging, but this has played a very important role. These are interdisciplinary networks involving scientists from many different fields and many places in the U.S. and in the world, and finally became a member of the Network for Socioeconomic Status and Health. And one of my close colleagues and collaborators is Teresa Seaman, who's a professor at UCLA, and you'll hear more about some of the things that she's done a little bit later. When this network closed, I became a member of the National Scientific Council on the Developing Child because it was quite obvious from the SCS and Health Network that many things that have lifelong impact develop early in life, and childhood adversity can have long-lasting effects, and you'll hear more about that later in the talk. As many of you know, the Whitehall study and studies in other parts of the Western world, the Whitehall study with Sir Michael Marmot in the leadership, have shown that there are gradients of health. This is mortality and this is morbidity across the range of income and education known as socioeconomic status. So people at the highest levels of income and education tend to have the lowest mortality rates and the lowest rates of many of the diseases of modern life. And there's a linear gradient. As one goes down to the lower end of income and education, you find an increase in both mortality and morbidity. Now, the reasons for this are multiple, and including the social environment, things like crowding and danger, the physical environment, toxins and noise, the availability of resources, food, transportation, recreation, the personal social environment, including the family, work and financial concerns, and health-related behaviors, whether we eat the right things, take in too much food, whether we are physically active, whether we smoke, drink alcohol, or how well we sleep. And we'll be talking about these things. This work led to a paper in New England Journal of Medicine in 1998, and which focused on the central role of the brain in mediating and perceiving what is threatening and therefore stressful, determining behavioral responses, determining physiologic responses, and all of these are influenced by individual differences, which we will definitely come back to. And then we consider the stressors. There's trauma and abuse, there are major life events, but there's also the daily hassles of our working, our families, our home environments, and our neighborhoods. And what the nexus of all of this is something we refer to as allostasis and allostatic load, and I'll be explaining that very shortly. Remembering that it's the behavioral responses, our lifestyle, as well as the experiences that can lead to a wear and tear on the body and the brain. And then finally, the National Council on the Developing Child has the focus on early adversity in life, childhood, and its long-lasting effects on the brain and the body, and we will certainly come back to that. So here are the main topics I want to cover. First, this idea of allostasis and allostatic load. Then what happens to the brain on stress, looking both at from animal models to people, top-down interventions, and then go to the topic of early life toxic stress, and finally talk about some important concepts to sort of summarize the whole our point of view. So if we look at blood pressure and heart rate, we know that it goes up when Somebody gets out of bed in the morning, walks up a flight of stairs, has an argument, gives a talk, as I'm doing now. But when these things are over, the blood pressure comes back down. Now, there are people who are hypertensive to begin with and have a further elevation during these kind of experiences. But it's this elevated level which can, over time, cause a wear and tear on the cardiovascular system. And Sterling and Iyer in 1988, and then with the late Elliott Steller in 1993, they first elaborated this, and we extended it not just to heart rate and blood pressure, but to other systems of the body that are involved in helping us adapt and survive. And we developed the concept called allostatic load, which means that when this hypertension or other elevation of these mediators of adaptation occurs, it's like a heavy weight on a seesaw. The seesaw remains in balance. Homeostasis is maintained for the time being, but over time the seesaw wears out, and there can be damage to the cardiovascular system and to other systems of the body. Now, when we think of stress, we often think of cortisol. And we know that cortisol secretion occurs from the adrenal cortex, from the stimulation of ACTH from the pituitary, and that in turn is driven by two peptide hormones, vasopressin and CRH, from the hypothalamus, and they are released with neural input from other parts of the brain in response to physical and also psychological stress. Now, when cortisol is secreted, it produces these biphasic U-shaped dose response effects. Acute stress can actually enhance immune function, can improve certain aspects of memory, can promote energy replenishment and better cardiovascular function. But when there is chronic or very high levels of cortisol, it can have the opposite effects. As we well know, it can suppress immune function and inflammation, impair memory, promote bone mineral loss and muscle wasting, and contribute to the metabolic syndrome. So we have these inverted U-shaped dose response curves. But we have to, and as an example of this biphasic effect, this is a study done by a former graduate student, now a professor at Stanford, Firdos Dabar, showing that when you put a rat into a restrainer for several hours and you measure the response to an antigen in an acquired immune response using delayed type hypersensitivity, this is the ear of the animal that is touched by the antigen and shows swelling, and this is the thickness of the ear. And you see that when you apply the antigen to the animal that has this required immune response, the ear gets thicker. But when the animal is stressed before this antigen is provided, you can see an increased thickening. And if the animal is immobilized and shaken in a gentle way in a shaker, there's an even larger stress response. And you notice it lasts for up to 6 days or even longer. So there's an enhancement of this acquired immune response, which is mediated, as it turns out, not only by cortisol, but also by adrenaline from the adrenal medulla, and by local cytokines and chemokines. Now, if you chronically stress the animal for up to 3 weeks, and then apply the antigen to the ear, here's the thickness, and you see the thickening of the control animal is here, but you see a profound suppression in the chronically stressed animal. So here, this acquired immune response has been profoundly suppressed by this chronic stress. And this is the kind of biphasic action of stress and stress hormones that we see in many